In our next section, we're going to finish up with some more organelles and then take a look at the cytoskeleton and then other things that are external to cells. In section 6.5, we're going to take a look at the mitochondria, the chloroplast, and peroxisomes. Now, mitochondria and chloroplasts are important in energy transformations, and they have some neat facts about them. They're not a part of the endomembrane system that we just discussed, but they are made up of membrane. They have a double membrane. And what's really interesting is that they have their own ribosomes and they have their own DNA. So the theory that's out there is that these were once prokaryotic cells that were engulfed by larger cells and became the first organelles. We call that theory the endosymbiosis theory. And that's kind of how we think that eukaryotic cells evolved from prokaryotic cells, is by these mitochondria and chloroplasts, which were once smaller prokaryotes being engulfed by larger prokaryotes and then just living inside of them. Now, taking a look at the mitochondria first, its main job is to perform cellular respiration. It takes carbohydrates, lipids, proteins, and breaks them down and uses that to create ATP. We're going to spend a lot of time looking at that process, and we're going to spend a lot of time looking at the physical structure of the cell, of the mitochondria. So on that previous slide, you'll see that the cristae are the folded parts of the inner membrane. So there's one membrane on the inside. There's an outer membrane. There's a space in between the two membranes that we call the intermembrane space. And then on the inside, we have the mitochondrial matrix. And this is where those ribosomes are located. As far as purposes right now, we don't really need to know the anatomy of the mitochondria. We will get more in detail with that when we look at cellular respiration more in depth. But your main focus for the mitochondria is that it does, quote-unquote, make energy. So it takes organic compounds, breaks them down, and uses them to manufacture ATP, our energy molecule of the cell. Our next energy type of organelle is the chloroplasts, and these are found in plants and other photosynthetic organisms. They're part of a member of organelles, or a class of organelles called plastids. Chloroplasts are different because they have different pigments. Specifically, chloroplasts have chlorophyll. Other plastids have other types of pigments. And inside of the chloroplast is where photosynthesis occurs. Chloroplasts are found in the green parts of plants. The most concentrated ones are in the leaves, but there are also some in the stems as well. Now again, just like the mitochondria, your text is going to go through and give you the anatomy of a chloroplast. We'll just take a quick look at it and then we will look at it more in depth later. There are two membranes, so we have the outer membrane, the inner membrane. Inside of the inner membrane are little stacks called thylakoids. They look like little pancakes, and this is where the light reactions occur, and we'll talk more about that of course later. But the thylakoids, when we have numerous thylakoids, they form a stack called a grana, and the stroma is the fluid very similar to cytoplasm that's inside of the chloroplast. And again, your purpose, the major function of the chloroplast is to perform photosynthesis. Our third type of energy-associated organelle is the peroxisome. Peroxisomes are similar to lysosomes in that they have enzymes that break things down, but these are very specific and they break down complex molecules such as fatty acids, alcohol, and in that process they end up making hydrogen peroxide as a waste product. Now, hydrogen peroxide, if we accumulated that in the cell, it would be pretty bad. So peroxisomes have also developed their own way of taking hydrogen peroxide and breaking it down, converting it to water and oxygen. That water and oxygen can then be used in the cell for other processes. Here's a picture of a peroxisome. Again, it kind of looks like a lysosome because it's a vacuole type looking thing, or a vesicle type looking thing. It is made of membrane, so it's very similar to lysosomes. So for our purposes, that's probably all you really need to know for peroxisomes, is they're just more digestive compartments. The next section we're going to take a look at is the cytoskeleton. And the cytoskeleton is kind of just what it sounds like. It's a skeleton of fibers on the inside of the cell, all throughout the cytoplasm. It's there for structure, it's there for support, and it's also there to kind of provide a roadway or a track for things to move along them, which kind of sounds interesting. Here's a electron microscope of the cytoskeleton, and cytoskeletons are made up of 3 different types of protein structures: microtubules, microfilaments, and intermediate filaments. Now, the roles of the cytoskeleton, as we said, it's there to support the cell, it's there to give it its shape, and it's also there to help things move around. So things like a ribosome or things like a vesicle can attach itself to the part of the cytoskeleton and use motor proteins to move along the microtubules or microfilaments, the components of the cytoskeleton. I do have a pretty cool animation that I will be showing you in class that shows you the components of the cytoskeleton and how this whole motor protein thing works. Now, as I said a few minutes ago, there are 3 parts or 3 components that make up the cytoskeleton. These structures are called microtubules, microfilaments, and intermediate filaments. Now, your text goes into pretty detail about the components and the structure and the function of each of these, but as long as you kind of realize that these are protein structures that are part of the cytoskeleton, that's probably all we really need to know about this. Now, microtubules are the largest. They have the largest diameter. They're made up of proteins called tubulin. Microfilaments are our smallest ones. They are made up of actin. And our intermediate filaments are ones that are in between, they're in the middle in size, and they're made up of different kinds of proteins like keratins. But again, that's probably a little bit more than you need to know. Now, in the cytoplasm, there are specialized microtubules called centrosomes and centrioles. These play a role in cell division, and we will see that when we talk about mitosis and meiosis, that these play a role in organizing the fibers that help to move the chromosomes around during cell division. So those centrioles and centrosomes are important in cell division. Another type of specialized microtubule is the cilia and the flagella. Now, these are actually external structures that help with cell motility, so they help the cell move, and they have similar structures in the fact that they are both made up of microtubules, but they're a little bit different. Cilia differ in their beating pattern, so how they move. They differ in their length. Cilia are typically shorter, flagella are typically longer, and on a typical cell, they usually differ in the number of them. Flagella, there's usually only one, and cilia, there's usually numerous. So here is an image kind of describing the difference between the two and some specific examples. Flagella, like we said, are usually longer and there's fewer of them. Cilia, they're usually shorter and there's more of them, and as you can see, there's a difference in the way that they move. In the motion, it's kind of more snake-like, and cilia, it's more of like rowing-like. Now, even though the cilia and flagella fall in the section of cytoskeleton, they're not technically cytoskeleton components. They're on the outside of the cell. And the reason that they included them in this section is because of their structure. They're made up of those microtubules that are also a part of the cytoskeleton. Now, the last thing we're going to take a look at are extracellular components, so things that are on the outside of the cell. We're going to take a look at the cell wall of plants, the extracellular matrix of animal cells, and then intercellular junctions. The cell wall of plants, as we've talked about, is made up of cellulose, those cellulose big carbohydrate polysaccharides. Also, there are protein and other things inside of them. And cell walls are also common to some prokaryotes, fungi, and some protists. Here's a picture of what the cell wall actually looks like. The cell membrane is on the inside, so the cell membrane is the inner layer, and the cell wall is on the outer layer. The cell wall is there to provide support and structure to the overall plant. Now, I believe your textbook goes in more depth with the primary cell wall, the secondary cell wall, and the middle lamella. We don't really need to know that for our purposes. Now, on animal cells, when you picture a typical animal cell, you probably picture that cartoon drawing that's in every textbook. But there is extra stuff on the outside of animal cells that they don't usually show in those cartoon drawings. And the extracellular matrix is that stuff that's on the outside. The ECM is made up of glycoproteins, so proteins attached to carbohydrates, such as collagen, proteoglycans, and fibronectin. And there's also receptor proteins that help recognize and adhere and stick to other cells and other things. Here's an image of what the extracellular matrix actually looks like. Here is our plasma membrane, so the double lipid bilayer. We have all of this other stuff on the outside. So we have proteins like collagen, we have proteins attached to carbohydrates, that's what's in green, and we have these integrin proteins, the ones that have the receptors kind of coming off of them. So as you can see, the extracellular matrix is quite complicated. On the next slide, what you're looking at is what actually is a proteoglycan, or what is a glycoprotein. Here you can see that a glycoprotein, like proteoglycan, is made up of both carbohydrates, polysaccharides, and proteins. So we have that core protein, the purple, attached to the green carbohydrates. So plant cell walls and the extracellular matrix are two external things that are going on outside of cells. Another thing that's going on the outside of a cell are these intracellular junctions. Now, think of a typical multicellular organism. They have lots and lots and lots of cells. And those cells organize to form tissues, and those tissues organize to form organs, and those organs organize to form organ systems. And they have to interact with each other, they have to stick together, and they have to be able to communicate with each other. Intercellular junctions are the answer to this. This is how they actually do that. There's plasmodesmata, tight junctions, desmosomes, and gap junctions. Plasmodesmata are in plant cells, and they are little channels that they actually go through the cell wall. So there's little tunnels that go from one plant cell to another plant cell. So on the top here, this is one plant cell, this is another plant cell, and these lines that are going vertical are the plasmodesmata. They're like little tunnels where molecules and signals can go from one plant cell to another. The next three types, tight junction, desmosomes, and gap junctions, are in animal cells. And the best way to explain this is to take a look at the picture. So as you can see, tight junctions are junctions or ways to stick together that hold the cell membranes of two different cells very, very tight. And that prevents any leakage, so things can't come from out here down in between the cracks of two cells. So it's kind of like a sealant. It's sealing the cells of a tissue together. Desmosomes, they kind of look like those little like anchors. They fasten cells together into strong sheets. So this is more of an anchor. We're not really sealing them off. And then gap junctions are connections that have tunnels as well. So these are very similar to the ones in plant cells because there's tunnels, there's channels that can go from one animal cell to the next. Okay, so that's kind of wrapping up all of the different parts of the eukaryotic cell. Again, make sure you're going through and filling out the worksheet 1 with the functions, the structures, and what they kind of look like in the cell.